We're at that time, aren't we?
Well, and
What was
yeah,
that?
what was that noise? Jenny, can you start that noise to scare me in the presentation?
That's the go signal.
Uh,
Okay, well, I think with that,
Welcome, everybody. Looking forward to the presentation today.
me
And,
too. We'll see where
yeah,
it goes.
Absolutely. Who's doing it?
I don't know. I think Jenny.
Okay.
All right. Well, again, thank you, everybody. So here we are. Somehow we're into June. I don't know how that makes sense. It doesn't make a lot of sense to me. But we're doing communication part 2. So I did this presentation, or at least part of the presentation, a little while back. And I wanted to kind of add to it before I did a bit more of a focus on plant communication. So now I'm going to do a little bit more focus on microbe communication and a little bit on how those plants and microbes interact, because, you know, I can't help but talk about that.
So do microbes have iPhones or?
I don't think they're exclusive to an Apple or Samsung product. They'll whatever's on sale.
Perfect.
Yeah.
Perfect.
And they do have little 5G antennae.
Oh,
yeah, that's what some of the cilia are. So just to kind of put in perspective and keep this in the back of your mind, it's a weird little idea, but nature is unbelievably sensitive and it's unbelievably complex. So there's a thing called silent mutations. When you're building a protein, you're using sets of 3 nucleotides, which is called a codon. And you have all these different sets of 3, these codons calling for specific amino acids. And sometimes you can have what's called a silent mutation, where here we can see we have a GAA, guanine, adenine, adenine. Here we have a guanine, adenine, guanine. So even though it's just an A to a G, they're still calling for glutamine. It's the same amino acid. However, even though it's the exact same protein structure, this little tiny change makes a difference. So my example is bun, patty, cheese, pickle, mustard, ketchup, lettuce. Bun is slightly different than bun, patty, cheese, pickle, mustard, ketchup, lettuce, bun. That little tiny pause right there, that extra tiny space makes enough difference that you can actually have a physical, a phenotypical reaction, and it can actually change the way the system functions. Pain sensitivity, as well as 50 plus other human disorders, are associated with a silent mutation. The protein doesn't change. The structure changes ever so slightly because of that small, tiny pause and how that protein is folded. So just in the back of your mind, keep in mind how incredibly sensitive these living systems are. So what is communication? And that is why we lift on 3. Communication is critical. We've got to pay attention. We have to have this idea of information. We have information sharing. And again, this is kind of a recap, so we'll go through it kind of quickly. But the idea of communication is information processing. We have to have an input of information, and then we have to be able to react to that information, have an output, and then feedback. And so we're seeing that in different species and different organisms in different way. And I have this color vision here. Humans we see in 3 colors, and then you have this mantis shrimp that it can see in 12 different colors, and it can individually polarize most of those colors. So what does it see? We don't know. We don't have access to that information. That's a big part of it, is accessing that information. And another way to think about it, like this one, if you were to take a pitcher and throw a ball at 100 miles an hour to a human, it looks like it looks just normal, 100 mile an hour pitch to us. However, if you were a leatherback turtle, humans can process at around 60 hertz. A leatherback turtle can only process at about 15 hertz. That's about 4 times slower. So that pitch that's 100 miles an hour to us, it seems like it's going at about 400 miles an hour to a leatherback turtle. And part of the reason it's so hard to hit a housefly is because they process at about 250 hertz, so about 4 times slower. So that pitch, 100 mile an hour pitch, seems like it's only going about 25 miles an hour. And it's all about accessing the world. And in order to do that, we have to have input. So it's talking about vision very briefly, where we have this visual input where we're able to process. But it's not just vision. There are lots of other senses, and I'm going to focus on one very briefly, and that's the sense of smell. inside of our nose, we have these little structures, kind of looks like a little hand. Hi, hi. And those are olfactory ganglion. And these are covered in receptors. They're just basically like a lock and key where proteins from the air, these scent molecules, these aromatics, go up into our sinuses and they interact. They lock into this shape. And it is incredibly specific. Limonene and pinene, C10H16, so same molecule or same formula. And in reality, looking at it, it looks like pretty much the same molecule. But these are known as enantiomers. They're mirror images like the left and right hand. And around this one atom, we have chirality. So we're setting up a left hand versus a right hand. And our nose is so sensitive that we can smell the difference, even though it's the exact same structure or looks almost exactly the same. We can smell orange versus lemon or lemon versus pine. And that's how sensitive these things are. And scent is a matter of opinion. Petrichor, the smell of rain, is typically a result of what are known as geosmins and a lot of actinomycetes, actinobacteria. Streptomyces are well known to produce large quantities of geosmin, and we associate that with the soil flavor, the terroir. However, just because we like it doesn't mean another organism likes it. And here we can see fruit flies. When they smell that geosmin, it is an innate aversion. They want to stay away. They don't want to oviposition. They don't want to lay their eggs, and they don't want to eat there. So a smell that we like is a smell that some other critters don't like. And I mean, flies, they've got a pretty weird palate, if you ask me. I've seen way too many out in my yard after my dog's been there, and whew. So the question becomes, do these communicate? I know that Dennis and I communicate, not very clearly most of the time.
I'm the leatherback turtle.
Yeah, in so many ways. But yeah, these organisms definitely communicate. And it's all about being able to access the information from the world around them. They have to utilize that input of information. But to talk about bacteria, we've got to have a little bit of the scale. Humans are huge compared to a bacteria. A human is 10 trillion human cells, and on our body, there's 100 trillion microbial cells. And inside of our gut, it's only a few pounds, but that's trillions of organisms. So to get an idea of it, the average human hairs, we're gonna call it 80 microns because it's easier for me to do the math. So we have to take that hair, chop it in half, chop that half 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 in half, 80, 40, 20, 10, 5, 2.5, 1 1/4. So we're down to 1 1/4 microns by chopping that hair in half and each half in half 6 times. That's the size of these microbes. So it's like splitting a hair. I know.
I actually used to do that with my hair and choose it as examples.
But you did it too many times.
Well, and that's why I quit doing it.
Well, it's all there. It's chopped up. Bacteria size. Yeah. So the question becomes, can microbes sense? Well, absolutely. So when we zoom in and we look at the surface of these microbes, we see all of these interesting structures that look a lot like those little fingers that we could see in our olfactory ganglion. And that's kind of what they're doing. They're out there and they're going to receive some of those scent molecules. So bacteria, at least the ones that can move, which are known as motile, they can move. And what many of them are doing is they're using either their flagella or their cilia, and they can twist those and make them move. And if they don't smell something that they want to go towards or something that they're afraid of, they can just kind of wander around. It's called their random walk. And then if they do smell something that they want to move towards, as you can see here, it kind of does a random walk and here and there. But as it picks up on this concentration gradient, it's able to move towards the food that it wants to move towards, which is very important when we start talking about the root ecosystem because of all those exudates and all those plant yummy foods that are coming off of that plant. So those microbes are able to move towards that food source, which is just incredible. and they're able to utilize that food, and that output can be pretty incredible. Vibrio fischeri is a pretty interesting example. So inside these little tiny, about the size of the end of your thumb, bobtail squid, they are a nighttime hunter. And being a nighttime hunter, there's sometimes a pretty major flaw in hunting at night when you have predators down below you, is you cast a shadow. So in order to overcome this shadow, these little bobtail squid house Vibrio fischeri in a specialized organ. And as these Vibrio fischeri grow and grow and grow, once they hit a certain concentration, they start to glow. And then this little bobtail squid can utilize those glowing organisms to simulate the night sky above, so that when a predator goes down below, it no longer sees that shadow. The bobtail squid has mimicked the light that's coming from the surface, but it doesn't glow all the time. The little bobtail squid is using those organisms during the night, and in the morning it spits out 75, 80, 90% of these little guys, and it stops feeding them. And then when it's time for them to start hunting and it's time for night, they start allowing them to glow again, and they do that by feeding them. So these organisms feed and feed and feed and reproduce like crazy, and then something kind of amazing happens. And that has to do with this idea of quorum sensing. Bacteria and a lot of other organisms have the ability to have an understanding of what's going on around them. Instead of a language, bacteria use signaling molecules, which are released into the environment, and these are known as the quorum sensing. And these can be intra-species, inside the same species, and there are different chemicals that they use for talking to their family, their friends, organisms that are like them, as well as interspecies. They can communicate with organisms that are not inside the same species, so they can talk to other organisms, and they can kind of listen in and have an idea of what's going on around them. Erwinia carotivora is a really good example of quorum sensing. And I mean, I know that my fridge has been guilty of having this happen, but zucchini, eventually they're just going to turn into mush. And they seem to be, and same with carrots, they seem to be okay, and then they're not. They fall apart very quickly. And part of that is because this organism, it sends out these molecules, these quorum sensing molecules, and it waits until there's enough of them. They slowly reproduce, they're sending out this communication molecule. And then all of a sudden, when it senses there's enough of them, it switches its genes on and it starts to attack. It emits virulence factors, which is, the disease part that shuts down other organisms, spits out enzymes that create a wound, an infection, and releases protective molecules to keep other organisms away. So it's waiting, and then sneak attack. It pounces. And that's going on with this idea of chit chat, this cell to cell communication. So we talked before about in the soil environment, there are a lot of these organisms that are producing bacterial biofilms, those extracellular polysaccharides that these organisms are using to glue our soil together. But they're doing more than just gluing it together. They're spitting out all these communication molecules so they know what's going on around them. And we've got all these different species, and they're all spitting out this cloud, and they can all kind of have a better idea of what's going on around them. And they can use this to coordinate their behavior. And these organisms are more than meets the eye. The litany of bacterial talents nibbles a conventional assumption about their thinking. These organisms, these bacteria can distinguish self from others. They can distinguish between relatives and strangers. They can sense how big of a space they're in. And they can also, this idea of utilizing this communication, they can hunt in packs. And this is oftentimes known as swarming. So here we can see a few different strains of Bacillus subtilis. These are all the same, but what's different is the type of food that they're feeding these organisms. And you can see their morphology, their shape changes considerably as these different species are exposed to different food types. And what we see is they have somewhat of plasticity. They're able to shift and change. And these pictures are Eshel Ben Jacob of Tel Aviv University. He did some incredible research and incredible work. And what he was doing, he was using mostly Paeonibacillus vortex, but he used another species or two. And all he was doing was exposing them to different food. And they would grow in these amazing, fantastical shapes. And that's just because of the food. And what he found is there are different types of bacteria that exist inside of a bacterial colony. Even those are the same species, they function different. And he compares this to different body parts, skin, blood, muscle, etc. And depending on the colony's needs, these bacteria will change. Keeping in mind that if temperature conditions are just right, these organisms can replicate every 20 minutes. That means that every time they replicate, they can change their phenotype. They can change how they're functioning. And his quote here, imagine every time you went to the gym, you could immediately convert your fat cells to muscle cells to deal with the challenge. Then you come back from the gym and maybe you want to read a book. So all those muscle cells turn into neuron cells. And that's what these organisms are able to do. They're able to shift and change and adjust to the environment around them. And it also allows them to kind of interact with different species. So this idea of the mycorrhizal fungi and bacteria, they collaborate. And a lot of these are known as mycorrhizae helper bacteria. So we can see these blue threads, these are our mycorrhizae, and around the outside of those mycorrhizae, we can see all these bacteria. If we think about the plant, the root is like my body, it's big in comparison. And then the mycorrhizal fungi hyphae, the threads are like my arm, and those bacteria are like the fingers. Now, if you're trying to pick something up and you're trying to extract something from an environment, you want a nice fine grasp. And that's what those bacteria are doing. They're able to extract more nutrition from the soil environment around them because they're just smaller. And that's just kind of the way it works. They're able to extract.
Kind of like picking a penny up off the ground with your elbows.
It's not gonna go well.
Yeah.
I'd like to watch it. I mean, even worse, imagine if like two people were like bonking heads trying to work together to pick it up.
Yeah.
So what's going on with these bacteria and mycorrhizal fungi is pretty incredible. They work together. But even across these different species, we have different abilities. So there's something known as the myco-CMN, Common Mycelium Network. That's the CMN. And a lot of scientists refer to this as the Internet of Plants. And scientists, being the nerds that they are, instead of the World Wide Web, they refer to it as the Wood Wide Web, which is just pretty stinking. It's cute. It's just cute. That's pretty fun. So what these organisms are doing is this is a collection of these mycorrhizal fungi that work together. And research has shown that plants that are part of the CMN have a higher likelihood of survival. That might be the same species of plant, but it could be different species of plants. So sometimes when we see an orchard where we're trying to establish a cover crop, by establishing those mycorrhizal fungi with our grass or with our fast growing crops, our crops that are in between our trees, we can increase the likelihood of those mycorrhizal fungi establishing with our orchard crop. So we can use cover crops as a way to do that handoff, because with mycorrhizal fungi, you want to have, it's like a relay race. You have to have that baton handoff. And so we can do that by utilizing different cover crops. And again, the presentation we had by Keith here this last time, incredible. So we've got to utilize all these tools at our disposal. And a lot of this research about the Common Mycelium Network started back with research by Susan Simard, and she was up in British Columbia, and she was doing research with Douglas fir trees. And so what she was doing is she had a theory that these organisms are able to transfer and move some of the food around. When you look at the forest, an extremely dense forest, you'll still have saplings popping up from the ground, and there shouldn't really be enough light for them to get established. And what she found is, well, they don't have enough light for full photosynthesis to allow themselves to grow. So what we're seeing is there's a transfer happening. In order to discover that transfer, she took different branches and surrounded some of the needles with just plastic bags, sealed it off, and then injected in radioactive C14, carbon dioxide with a radioactive carbon. And then she monitored the uptake in the tree, and she could see it going in through one tree and then being spread down through this common mycelium network around to other trees. And that's where this term mother tree came in. These big giant trees that are out in the environment are playing a very critical role in the health of the other organisms around them. If you take out one of these mother trees, it has a much larger impact than a tree that isn't one of these highly interconnected trees. And you start to lose some of that protection and some of that food transfer if you take out some of those very, very large old trees. And it goes even deeper. So we're talking mostly about chemical communication and some very recent research. This actually came out in April 2022. Andrew Adamatsky, UWE Bristol, was doing research. He's actually a computer scientist, not really all that much into biology and botany, but he had some theories that these organisms are able to do a bit more than we originally thought. It's not just chemical communication. What he found is through electrical impulses, much like our brain uses, these organisms are able to kind of speak to each other. At first, kind of assumed it was just random spikes of electrical impulses, but the reality is he started to see patterns. And he found that by monitoring these different fungal species over time, he saw that they kind of reuse these repeating patterns, and some of them are using up to, he's calling them words, up to 50 words. And on a regular basis, they're using 15 to 20 fungal words to communicate. And they think they're probably utilizing these chemicals to talk about threats in the weather. Beware of humans ahead, danger, look out. Oh, it's good weather for us today. So this fungal word length is about the same, according to Adamatsky, as ours. They're about 6 characters long. And in English, our words average about 4.8 characters long. So very similar. Shocking, isn't it? Absolutely. Different species have different electrical spikes. So they kind of have their own distinct communication. And like I said, these spikes are very similar to how our brain works. Our brain, our neurons send pulses from one neuron to the next, and it's an electrical impulse, much like making your muscles fire. Your actin and myosin, which electrical impulse is causing to basically grow along each other. They stretch along, and that's how your muscle flexes up. And that's just electrical impulses. These fungi are able to do a pretty similar thing. And repetition of patterns could show purposeful interaction and communication, which is just incredible. And whenever we start to think that these are just small, dumb organisms, I always like to think about these cordyceps. Don't underestimate the power of these small organisms. So cordyceps, here you can see, this is a rainforest. I believe these are down in the Amazon. And the cordyceps will basically utilize these ants to spread themselves. So for the cordyceps to have optimal growth and optimal spread of its spores, it wants to have a very specific environment. So it requires the north-facing leaf of a plant, about 25 centimeters off the ground, in an environment with 94 to 95% humidity, in a location with a temperature between 20 to 30 degrees. So what the cordyceps does is it infects the ant, it grows into its brain, and it hijacks the ant's central nervous system. It forces it to go to this perfect environment, which is usually above where the rest of the ants in the colony are wandering and walking, so that it can rain these spores down and spread. That's how precise the control of the fungus is. It can navigate an ant to the absolute perfect location down to a single percentage point of humidity. That's not just ants, they're affecting other species as well. But I don't want you to think that just because we've got a few bad apples, they don't spoil the bunch. I know, it's pretty cheesy. So when we start looking at plant growth promoting rhizobacteria, PGPR, we see a lot of benefits and impacts. And this is where we can see some of those benefits directly to the plant. These are powerful allies. Biofertilization accounts for approximately 65% of the nitrogen supply to crops worldwide. And I think we can increase that by utilizing biological growing practices and a lot of the nitrogen fixing bacteria. Beneficial soil bacteria confer immunity against a wide range of foliar diseases by activating plant defenses, thereby reducing the plant's susceptibility to pathogen attack. And we'll talk a little bit more about that. But these small organisms can make a big difference. I've shown this picture quite a few times before. This was in New Zealand. This is an established fescue. This is the same area, within a meter, same grass 3 weeks later. And you can see the growth stimulation. that's happening. And these microbes, they want to help the plant grow. The more the plant can grow, the more it can photosynthesize. And the more it can photosynthesize, the more sugar it has. And the more sugar it has, the more it can spread that wealth around. So research shows that about 25% of photosynthetic sugar is sent down and out through the roots to feed these organisms. And at certain stages, you can have up to 75%. Saplings and seedlings, they'll send 30 to 40%. These babies that are barely able to survive are still sending that much food down and out. And it's not random, it's purposeful. Dennis talked a little bit about Jill Clapperton's work at looking at different profiles of the exudates on different plants. And there are quite a few different molecules. It's not just sugar water they're dumping out. They're doing this on purpose. And they're recruiting these organisms in. We're not even going to talk about rhizophagy cycle right now because, well, we don't have time and we've covered that, but keep that in mind as well. These plants are attracting in the right organisms. So here we can see a number of molecules that are used for communication, but I'm focusing, I underline here, malic acid. And malic acid can be produced by the plants and it attracts in Bacillus subtilis. So here we can see research was done. The professors threatens this plant with Pseudomonas syringae, and then they monitored to see what would happen. The plant started spitting out lots and lots of malic acid, which attracted in the Bacillus subtilis, which they've radio labeled, or they added the green fluorescent protein so that it would glow. And all this green around that root is all the Bacillus subtilis. So even though that syringae is out there in the environment, it doesn't have a whole lot of access to those roots. But this communication can also kind of backfire if we're not paying attention. If we're doing it right and we're adding diversity and we're adding different species, we're allowing more of this communication to happen. But sometimes humans, we don't really know all of what we're doing. Remember those silent mutations and how precise the control is over these biological activities? A 2011 report found that commercial corn hybrids seem to have lost the wild maize plant's ability to release chemicals that attract parasitic wasps that kill stem borer moths. Bruce, years ago, when he was a plant breeder geneticist, he focused a lot. He spent a lot of time and a lot of research on trace mineral uptake. And when he was developing new seed lines, he'd make sure that he wasn't breeding out trace mineral uptake capacity, because trace mineral uptake is critical for the plant's function, for its proteins, its enzymes, and its immune system. So when we don't pay attention to these things, we can lose things. And we need that collaboration. We have to have it. These organisms, even though they're small, they can do some pretty incredible things. Altruism is the idea of one organism sacrificing itself or decreasing its own health for the benefit of others. Tell me, Carl, are you familiar with the expression, take one for the team? In studying the development of antibiotic-resistant strains of bacteria, researchers found that the population most adept at withstanding doses of antibiotics are those in which a few highly resistant isolates sacrifice their own well-being to improve the group's overall chance of survival. And in this case, what he found was, I think they were studying E. coli, they were sending up indole. It's a signal flare. When the organism, these slow growers, and they're kind of purposefully slow to grow. As you increase your rate of growth, you're more likely to take up more antibiotics from the environment. So purposefully, like Dr. Ben Eschel talked about, where we have these different organisms inside the same colony having different functions, these organisms are slow growers, and they're able to kind of keep an eye on things. They monitor the environment, and when they sense an antibiotic or a threat, they'll send up this signal flare. That encourages a biofilm and stationary phase. It hurts or costs the individual its health and its growth, but it helps protect the overall colony. Over 85 species have been found that single shot approaches may miss quote unquote sleepy individuals. And that's where a more biological approach can be beneficial. They're organisms, they're growing, they're with the plant, they're with the species, they're interacting and working with the environment versus chemical approach where sometimes you can miss. And how many different weed species are we seeing that are resistant? Lots. And that's because some of these are a little bit slower and they're more purposeful. And we see the same with bacteria. Evasive maneuvers, bacteriophage, and here you can see some cool bacteriophage. I think that they're one of the coolest looking little critters. They're a virus, and what they're doing is they're hijacking the bacteria and the bacteria's own internal machinery to reproduce themselves. So again, we can see when some of these organisms sense the bacteriophage, rather than threatening the rest of the colony, they'll sacrifice themselves. And through a process known as apoptosis, which is programmed cell death, these organisms that have been infected by the phage, they don't want to reproduce and spit out millions of them. They allow themselves to die. And when looking at different species, even inside of different species, you have different traits, different characteristics. And here you can see a population where there's a lot of clear on the plate. And that means that the bacteriophage has wiped out a lot of these bacteria. And here we can see a very similar species, but this one is more adept at shutting itself down, utilizing that programmed cell death to stop the spread. And we have very few casualties. Vibrio, I mentioned the Vibrio fisheri before with the bobtail squid. This is another Vibrio and another characteristic of the Vibrio. And here we can see this massive interaction, all these individual lines, these individual spikes, those are different species of the Vibrio. And the way they interact can be incredible. You can see this one like up here. It has a massive number of interactions with a wide range of different species, subspecies inside of the Vibrio. And Vibrio have been found to protect each other. And they can produce antibiotics that are very specific. They can produce antibiotics that harm their relatives and those that are more closely related less than it harms other organisms. So before we talked about those quorum sensing molecules, they have the sophistication, the ability to recognize close kin. And they don't want to hurt them. They want to help their species survive. So the antibiotics they produce are targeted towards, even though they're all Vibrio, different Vibrio. Getting in those genes. So these organisms are able to share information. Not going to go a whole lot into genetic sharing. I wanted to point this out. Bacteria that have antibiotic resistant genes advertise the effect by attracting other bacteria shopping for those genes. Later then, they emit pheromones to signal their willingness to close the deal. So organisms that have the ability to resist that antibiotic, a lot of times they'll package that information, that DNA, that ability to protect themselves in what's known as a plasmid. And then when they basically advertise, like it says, the researchers are finding, they advertise their ability to resist that attack. They'll let others know, and then they'll share that DNA, that ability through a plasmid, which is just incredible. We're also seeing that when the plant is thirsty, these organisms are able to help and benefit the plant. So this is the idea of soil signals. During a drought, abscisic acid is produced, which translocates to leaves to regulate the stomata. Soil microbes help close the stomata and help the plant conserve water. Not only that, there are organisms known as potassium mobilizing bacteria. And these potassium mobilizing bacteria have a substantial effect on enzyme activation, protein synthesis, photosynthesis, stomatal movement, and water relation. So they're able to communicate with the plant. It's starting to get dry down here. They're able to increase the abscisic acid, which allows the plant to more easily open and close its stomata. Think about like shutters when it's bright out. And the combined effects of low temperature and low moisture can be alleviated by increasing the concentration of potassium in the soil by these organisms. So working together, these organisms are able to help protect the plant from what are known as abiotic stresses, drought and salinity, even heat. And these PGPR, we've talked a little bit about this before. We're not going to go into a lot of detail, but these organisms have a wide variety of impacts and a wide variety of chemicals that they're utilizing to help communicate with the plant and protect the plant. ACC deaminase is one of those. When a plant is exposed to drought or salt stress, it produces a lot more ethylene. Ethylene is the hormone and the communication molecule that encourages the plant to hurry up. We got to get to the finish line. We've got to produce. So these organisms like we see up here, have a wide variety of benefits. They can help the plant. They can help the plant grow. They can help the plant extract nutrition, and they can help the plant decrease its stress, which is an incredible ability, especially when you start looking at some of the environments that we're growing in, some of the ag areas we're growing in, that they don't have great water. And this is a way that we can help those plants grow through some of that stress. Another interesting way that these organisms are impacting the communication and the chemical responses of plants is through modifications to allelopathy. Allelopathy is basically plants spitting out molecules that suppress the growth of others. Some of them do that directly, some of them do that through volatiles. However, others require these organisms, require these bacteria, these PGPR in the soil to modify the chemicals they're spitting out. And they don't become fully active until these organisms have modified those chemicals. And when we start looking at the responses of the plant, its immune response, I say talk isn't cheap. And that's because every time a plant needs to do something, it has to spend energy. It has to utilize its sugar to create new molecules, to create that communication. And that's where sometimes the bacteria can come in and be beneficial. So this is massively oversimplified, but we can think about abscisic acid as primarily as abiotic stresses. Osmonic acid is primarily herbivory, insect attack. Salicylic acid is typically associated with disease attacks. The plant is going to upregulate or downregulate some of these hormones based on what's going on. Systemic acquired resistance, it's similar to our resistance. It's like a flu shot. Generally, systemic acquired resistance is associated with a plant gets sick, it either makes it or it doesn't. If it makes it past it, then it has now figured out how to fight and protect itself from that disease. Researchers have found that when plants were treated with a PGPR, Bacillus pumilus in this case, they were able to increase their salicylic acid levels, which allowed them to kind of upregulate and prepare themselves for the attack so that it's like they know it's coming and they can defend themselves more quickly. Another way that they're able to do this is through what's called induced systemic resistance. Term induced resistance is a generic term for the induced state of resistance in plants triggered by biological or chemical inducers. Basically, these PGPR, these organisms down in the soil, are able to let the plant protect itself more easily. It's kind of like queuing it up so that you're on deck. It's like playing baseball and you got your batter on deck. You know that it's about time. You know that you need to protect yourself. You know there's something in the environment. And again, they're doing that with these communication molecules. I like to point this out because this is the current state of understanding. There's still question marks. We know that there are compounds and molecules that these PGPR are producing, and we know that they impact the plant's growth and health, but we don't fully understand it yet. And I think that's exciting because that, like Adam Atsky's work, we're finding out new things. And as we utilize and understand more of this, we can be more targeted with some of the beneficials that we use, and we can allow them to help protect the plant from different things. Here's induced systemic resistance against cucumber beetle. Again, this is a plant growth promoting rhizobacteria, a root organism that they're applying to the root of the plant that helps protect it from beetles. Here we can see again, cucumbers treated with PGPR. I believe this is a Bacillus pumilus in this case, and they're helping protect from bacterial wilt. And I just found this research. I thought it was pretty interesting. And here we can see, again, researchers in this case are utilizing Pseudomonas fluorescens, and we can see these are treated leaves, these are untreated leaves. And as we look at the zoomed in, these blue lines, those are the fungal hyphae. So they're there, they are growing, they are present. However, comparing D to E, there are a lot more of these little round balls along these blue fungal hyphae. And these round balls are known as haustoria, and haustoria are basically the site of extraction. A while ago, I talked in a presentation, I'm not going to get into it, but this haustoria is actually a site of genetic exchange as well, where both of these species are exchanging genetic material. And it's more exchange than researchers would think would normally happen. So there's more going on there than we originally gave credit to these fungal mycelia. But having less haustoria It's like punching fewer holes. So yes, this fungal pathogen is present, but it does less damage. And again, that's utilizing PGPR in the root to help protect the plant above ground. Let's see, is this video going to work? Oh, here we go. This is kind of a fun one. University of Delaware. And here we can see this is an untreated plant. Here's the stomata in red, and these are Pseudomonas syringae. And whoop, there goes one right inside that stomata. Oh, there goes another one. That plant's now infected. It now has Pseudomonas syringae inside of it, and they're just free to go in and out. Here, the plant was treated with probiotic. This is a bacillus, I think Bacillus pumilus again. And here we can see the stomata is closed, and these pathogens can no longer make their way in. The PGPR, the soil organisms have communicated with the plant. There's something on the way. You better protect yourself. So the plant has and does. Oh, what happened? There we go. And it's more than just direct communication, a lot of hot air. So there are things known as microbial volatile organic compounds, VOCs. And I talked about VOCs before, but these microbial VOCs have a strong and large impact on a wide variety of different organisms, fungi, bacteria, plants, protists, other organisms that are in the soil. Research has revealed that without physical contact, microorganisms are able to drastically alter plant root system development, plant physiology, hormone pathways, and biomass productions. These microbial VOCs can also function directly as a food source. So not only does the plant use it to communicate and have a better understanding of what's going on around it, it says, thanks, now I have a better idea. Now I'm going to also utilize that as food for myself. These organisms that are down in the soil are respiring. They're doing their normal life activities. They're breathing in and out. And that CO2 that is generated also directly feeds the plant. So utilizing these organisms and the gases, this is just the gas they're spitting out, induces resistance to pathogens in plants. A plant affects plant secondary metabolite production. It can increase secondary metabolite production, allowing the plant to better protect itself. It can directly inhibit plant pathogens, and it can induce soil fungistasis and suppressiveness. So just these the breath out of these microbes, these PGPR, is able to slow down the growth, the virulence, and the activity of fungal pathogens. And we can see that with induced systemic tolerance. So by having these VOCs in the environment, the microbe is able to communicate with the plant and help protect the plant from drought and salt stress. So utilizing this, and we saw all those other benefits, the ACC deaminase, the osmolytes, there's a lot going on with these microbes directly through communication and chemical release, as well as just through the VOCs, the gas that these microbes are producing. So call this alphabet soup, PGPR, ISR, VOC. Whew. Those are plant growth promoting rhizobacteria, inducing systemic resistance by utilizing volatile organic compounds. That's a mouthful. That is alphabet soup. VOCs significantly upregulate the expression of genes related to wilt resistance and pathogen defense. Even if the organism isn't directly interacting with the plant, it can still have benefits. And this is where, when we have a monocrop in a field, or we have decreased diversity in the field, we can utilize some of these organisms. Whether or not they're the best species to work directly with a plant, they can still be producing some of these VOCs. They can still be communicating with the plant, whether that organism is directly connected to the plant or not. Simply by having the organism there, we change the environment and we allow that plant to more fully express its genetic potential. And that's where, repeated application of some of these organisms can be beneficial. If we have drought stress, if we have waterlogged soils, if we've done something to disrupt these communities, that's where coming back in to allow them a chance.
Kind of like your baseball analogy, you got the batter on deck. Well, sometimes that environment changes and they want to throw in a pinch hitter.
That's right.
If that pinch hitter isn't on the bench,
Yep.
They can't utilize
Yep.
it in order to win a baseball game.
Yep. You've only got access to the tools that are in your disposal. If we don't have the tools, the microbes, or the tool in this case, in the field, plant can't use them. And that's where biological farming, regenerative farming makes a huge difference. And again, we're just barely starting to understand all the benefits. Callose is a plant sugar. It's a beta glucan. And here we can see, I like to think about this, it's kind of like thick gloves. Your skin is kind of strong, but what do? You put on gloves when you got real work to do because you don't want to tear up your hands. So this callus can be deposited by plants to help protect itself from these fungal mycelia. So if the mycelia penetrate in and there isn't much callus, they've made it in, and now they can start accessing the nutrition from the plant. However, if the plant has increased its glove, its thickness, its protection with this callus, then it can basically stop that mycelia from penetrating more. And in addition to forming a physical barrier for pathogen penetration, callus depositions are also considered to be a matrix for the accumulation of defense compounds, such as hydrogen peroxide, phenolics, and various proteins, glycoproteins, the hydrolytic and antifungal properties. So by having PGPR in the soil, we allow the plant to have access to more information, have a better idea of what's going on in the world around it, as well as, literally thicken itself. They're able to deposit more callus when they know they need to. Again, remember, everything is an energetic balance. A plant doesn't want to have to do this. But if it senses there's a threat, if it senses there's some sort of disease, it wants to protect itself. So this is one of the things that it can do by utilizing all these PGPR, all these beneficial organisms that are in the soil and around it. So yeah, again, here you can see with these PGPR, these beneficial organisms, we can induce this state and we can increase our callus deposition. So I wrote down same destination here. So we're ending up at the very end of this with systemic acquired resistance or induced systemic resistance. And we talked about how these PGPR can help protect the plant, either directly SAR or indirectly ISR. And we get there basically the same way. We can have pathogens, chemical attack, chemoactivators that are allowing this exchange of information, this immune response, or we can have PGPR. We can have these signaling pathways from these beneficial organisms into the soil, into the plant, through VOCs. All this communication can be going on to allow the plant to do the same thing, to help protect itself and grow. So the assumption of nature's communication is this. You got a few switches, not much is going on. Is that a Nash?
Is
that a Nash Metropolitan?
Yep.
Oh, yeah, look at
That's
that.
a Metropolitan. Yep.
Yeah, pretty good one. The reality is, oh my goodness, there is a lot going on. And just like me, I'm no pilot. I don't read and understand all that information. I guess it's like the fungal spikes. I don't speak that language yet. But as long as we're paying attention, we continue the research, we can start to get a better idea of what all these different buttons, bells, and whistles can do. And once we have a better understanding, we can utilize them. In the meantime, we don't have to fully understand them. We just have to get those organisms out there because nature is going to do a lot of that for us. So I have here the takeaway: respect the. We'll talk about skin cancer and how some beneficial organisms can protect you from it later. Respect the beauty and intricacy of nature because all life on Earth is extensively interconnected. Nature speaks in a language that we are just starting to understand. We are working with living organisms, so we need to not have a reductionist mechanistic view. We need to have a holistic view, and that's where regenerative agriculture, biological farming can come in. And again, a few decades from now, we're going to have a better understanding of all the things that we've been doing and all these benefits that we've been reaping from having a diverse biological community in our soil. For now, we just get to have the benefit. We don't fully understand it, but we know good things are happening.
But if you think about it, where this technology and information has come from 30 years ago to 15 years ago to 5 years ago.
It's incredible.
And
leaps and bounds.
Leaps and bounds. And, I kind of give the example, and I'll go ahead and use this here. It's kind of, I remember the old Charlie Brown when it came out. And, and the teacher used to talk, it was wah, 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 wah. I mean, that's kind of what we hear and understand. Now I know the teacher was saying, you're a blockhead, Charlie Brown.
Well, I'm a parent. I've got a kid, and all my kids, all they hear is wah, 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 I feel like half the time. And I'm the blockhead still.
Well, yes.
Okay, with that, I think, let's see what sort of, let's see if I can open up the chat and find some questions. Sorry, give me 1 second to kind of read through some of these.
You know, so much of what we see in agriculture on a day-to-day basis ties into everything that you talked about today, Steve.
Oh, thanks.
I mean, yeah, I mean, when we start to talk about our row crops and our mycorrhizae fungi
Yep.
within, and
Yep.
that interconnecting or that cross infection from plant to
Mhm.
plant to plant,
Mhm.
and we see those mycorrhizae helper bacterias. And, you know, a lot of times when we're talking with growers, we talk about the idea of, well, I was more susceptible to mildew, powdery mildew, downy mildew, but I didn't get it. And we always talk about the benefits of nutrition.
Yep.
The benefits of a healthy plant,
Yep.
a healthy soil. Those are all the things that we now are starting to see being expressed
Yep.
in field environments. You know, some work that's going on in weed control that
Mhm.
we're doing in some different areas from the standpoint of just through nutrition
It
and
changes the profile.
profile.
Yep. Absolutely. I mean, there's no doubt there. Some of the books, Charles Walter's
Yeah.
books and weeds and what they tell us and all those, there's a lot there. The environment is critical. The environment dictates who is going to be the dominant player. One of the problems with glyphosate is that it also hits the viability of some soil microbes. Subset of these could have beneficial potential for the plant. Oh, absolutely. Dr. Huber, Dr. Kremer have talked about that for quite a while. Pseudomonads are one of the ones that they've kind of singled out and talked about. And we know that Pseudomonas fluorescens, Pseudomonas butida, there are a lot of benefits that we're getting off of those organisms. But as we apply some of these chemicals, it shuts those species down. It can have a very hard hit on some of those. In those cases, they point those out because they're reducing organisms to have most of your elements. It's like iron versus rust. One is functional, one is not functional. And all it has to do is the oxidation state. Same with manganese, same with pretty much all of our trace minerals. If we don't have it in a biologically available form, the plant can't utilize it. You know the best way to make it biologically available? Pass it through biology. Send it through a microbe. And then we're gonna get there. Okay, let's see. Good point. Oh boy, Kathleen, I think we're gonna need a little more information. Last season, I had a fungus develop throughout my garden. Now I'm seeing brown spots starting on some plants. What does this indicate about my soil? What will help balance my soil? I am currently spraying AA's Vitality and Growth Weekly. We're gonna need a bit more information. We've talked about many times before, test, don't guess. We're going to have a better understanding of what your nutrition looks like, what your plants look like, what sort of species you've done before. If I do tomatoes over and over and over again in the same environment, I've got to break that disease cycle. If I'm utilizing some plants that they're very well known to pass that disease from one generation to the next, we got to break that. So there's some kind of simple things we can do. We can look at our watering practices, our water quality. Basically, what I'm saying, we need a whole lot more information. We'd love to hear more of that information from you. And that's where it's a good reminder. That's a great place to start. Leaf extract analysis, work with apicals, soil analysis. This data is going to be critical to helping us understand more of what's going on.
Because so often, I mean, you go back to the idea, Steve, that it's soil health, it's microbial communities within that soil health, getting those PGRs functioning, but we also have to have those exudates and a healthy plant to get the food down into that environment. So kind of taking a look at the whole package there and being able to give you some ideas of which direction you might want to actually go.
Well, and it can be a challenge depending on the soil. I remember a while ago, in a presentation, it was the question of, am I using PSB or the drought stress, and those roots on the mint plant?
Yeah,
they were non-existent. If we're trying to get nutrition in through the root, we don't have much chance. So there are times where putting nutrition in through the foliage is going to be the best way to get our biological communities in the soil back up and running. So we've got to have a lot of information to know. Let's see, some plant families do not respond as well as others to being inoculated with mycorrhizae, including beneficial soil bacteria blends. But are they injured by inoculation? Yeah, and that's a good question, good point. 95% of all species make a mycorrhizal connection. Most of them are arbuscular mycorrhizal fungi. 85 to 90% of species are working with those. You've got ectomycorrhizae that work with a different subset, iricoid mycorrhizae that work with a different subset, and then you've got some brassicas and others that, as they understand it now, do not make that connection with mycorrhizal fungi. Do the mycorrhizal fungi harm? No.
Yeah.
They just don't do anything. They're not going to have much interaction with them.
Well, and that goes back to the idea of a cover crop that I have seen a lot of
Yes.
times where you have a plant that will not create a
Yep.
relationship from mycorrhizae, but we have a cover crop scenario there that
Mhm.
does. That actual crop that doesn't can get the benefits from exudates
Yes.
and
That's
that
it.
nutrition being
Yep.
broke down within that environment just by having those there.
Yep.
Yeah.
The more of those species we have, and sometimes we have like an orchardist that's nervous about putting cover crop in between their trees, their rows. If the trees are just barely getting enough nutrition that they need every bit that they can get, sure. I'm a little nervous during that transition. We might end up with a small lag when those organisms are sucking a bit of nutrition before they're spitting it back out. But if we have adequate nutrition, if we have adequate carbon levels, if we're allowing those organisms to do their job, that's what they're there for. Looking at Dr. White's research on rhizophagy, a lot of plant species have to, and I mean have to have those beneficial organisms to grow normally. Without them, they don't develop root hairs, they don't develop normal roots. So we have to have those species. The alternative is basically putting the plant on life support. You're doing salt-based fertility, and you've got to have it dead on. If we extract and we don't utilize the plant's digestive system, which is the microbes, the plant's gut is basically outsourced to microbes. If we bypass that and we don't focus on that, then we, as the grower, are responsible for every bit of nutrition. And if we make a small mistake, plant lets us know. Biology is a buffer.
It is a buffer. And I a lot of times look at competition is we only have competition is if we don't have the habitat in order to support that environment.
Yeah.
And so a lot of times we have to look at that as growers is how do we support, because it really comes down to what is the best way to build that biological community? So nutrient is actually being made available to that plant. And a lot of times I see that in the tree industry is we don't want to have competition, but we also want to do everything we can because we're setting up that plant for the next 20 years, 30 years.
Yep.
Yeah.
Oh, definitely. Vineyard plant protection, foliar fungal disease. Should I apply protected bacteria to soil for best protection, or will I get better protection from foliar applications? Oh, Greg, I think we're going to need more information. And again, there are different products for different application. Different application types. None of the Tainio products, we're not control products. We're not EPA registered. That's not the intent of our product. This information is just information to make everyone more aware and more up to date on what's going on and what the capacity of these organisms is. So generally, if we're fighting a foliar disease that comes from the foliage, then we want to do a foliar spray. However, there are times where the source of a lot of those fungal pathogens is actually the soil. Damp soil, litter on the soil being decomposed, and then creating those spores that end up on the leaves. So in that case, we've got to focus on usually the soil first. By the time it's made it to the plant, we've kind of already missed a window. We've already missed a bit of an opportunity to slow it down.
It goes back to the idea of program and be proactive rather than reactive when it comes to this type of thing, and trying to develop the best set of tools within that environment in order to outcompete.
Test, don't guess. You're never going to have an environment where those diseases aren't present.
Correct.
They're always there. It's just whether or not they can get a foothold in the plant and impact the plant. Back to rhizophagy again, some of those, or at least the work by Dr. White, some of those species will actually, those PGPR will actually infect the fungal pathogen and shut it down. They can digest it, they can slow it down, they can reduce its virulence, and in some cases, they can actually make it beneficial to the plant, which is just incredible. There's so much going on. It's here, let's see, I'm going to go back up to that one. There's so much more going on than we give it credit for. I think We made it through.
Yeah.
Questions. Look at that.
I
An
know it.
hour. That
An
is the
hour.
shortest one
That's-
we've done in
That's
a
a
long
record
time.
time.
I know.
Record.
Our half-hour presentation only lasted an hour.
Well, and any of the questions you have out there, you can always
Yes.
email us directly at info@Tainio.
Yep.
Steve or myself,
Mhm.
and we'll be happy to answer these questions any way we can, based off of giving a little more information.
Yeah, Kathleen, that same for you. Send us some information, get us some analysis so we can take a look. Greg, same with you. Let's take a look and see what's going on. A doctor that makes, and I'm not saying we're doctors, a doctor that makes a prescription or a diagnosis without any information is not a doctor that I would want to go to.
Yeah.
You've got to have a lot of information to be able to have a better understanding of what's going on. What's the causative agent? What's behind that? We've got to dig down until we find the cause behind the cause and able to treat it. And it's almost always nutrition. If we could have every crop have adequate calcium and phosphorus,
Yeah.
a lot of our problems would disappear.
But
But how do you get calcium and phosphorus?
That was, you knew where I was going with that, huh? Maybe there's some beneficial bacterium and beneficial fungal that might help with that.
Are there
a little
any
bit.
like phosphate solubilizing bacteria that also solubilize calcium? Oh, like the Spectrum PSB.
Wow, only if they're tied up together. Does that ever happen?
All right, our sarcasm needs to be done.
Thanks, everybody.
Thank you
Really
so much.
appreciate it.
We had a lot of fun. Hopefully you did as well. And we'll see you next time for, I don't know who's doing what, but we'll have a lot of fun.
That's right.
Welcome to June. We're getting closer to summer. Thank you, everybody, and talk to you later.